So first, let's go over again a little bit of comparing the two of those processes together. Big picture stuff. Number one, mitosis is going to conserve the number of chromosome sets producing cells that are genetically identical to the parent cell. And that's why we said you start with 46 as the parent cell, the daughter cell will also have 46. And it's a clone, it's identical. Meiosis, on the other hand, it is going to reduce the number of chromosome sets from 2 to 1. So we go from being diploid to being haploid. And that produces cells that differ genetically from one another and from the parent cell. So every single one of those gametes, those sperm and eggs, will be different from each other. But we could say a similarity, if we're comparing the two, is the fact that if you look at the steps of meiosis 2, they're identical to the steps of meiosis 1. Of mitosis. So meiosis 2 is exactly the same as mitosis. The prophase, the metaphase, the anaphase, the telophase, no different. Okay, it's just the meiosis 1 then that you really gotta focus on and learn those steps well, and all those special things that happen during meiosis 1. Okay, I'm going to zip through this real quick here and get us here. So what we're going to talk about for a good portion of time here, again, is how you become so unique. We'll look at a variety of ways that happens through meiosis. But for this slide right here, let's just take a look at some of the big things that go down in meiosis 1 that make it so special. Okay, so number one, the process of synapsis and crossing over. Okay, so synapsis, again, that was when initial part of prophase 1, where your homologous chromosomes, they're all in the nucleus, and there's 46 of them floating around, and almost magically the individual chromosomes of the maternal and paternal side from mom and dad find each other. Chromosome 1, chromosome 2 finds each other. The 2 from chromosome 3 find each other. The 2 from chromosome 4. And that takes some time. That's why prophase 1 is the longest stage of them all. And then we said that once they do find their partner and they're sitting there floating in the nucleus with them, well, then they do that unique thing called crossing over, where the 2 non-sister chromatids in the middle essentially start wrapping their chromosomal arms and legs around each other, and they break, and when they finally separate, you'll notice they've exchanged portions of those chromatids. Another big thing that happens during prophase 1 is the events of metaphase. Okay? Now, metaphase is usually people's favorite phase because it's the easiest one to see. All the chromosomes are lined up down the middle. But in meiosis 1, it's a little bit more organized because your homologous pairs of chromosomes, the one from mom, the one from dad, for all 23 pairs, find each other and wind up at that metaphase plate. In mitosis, it's just kind of every chromosome for themselves. They all just kind of float to the middle, and there's not really any organization there. It's just very, very random. But in the case of meiosis 1, they go there with their partner. The maternal chromosome goes there with the paternal chromosome. And then at anaphase 1, another huge thing that happens in first round of meiosis is the fact the homologous pairs are the things that separate. In mitosis, during anaphase, it was the sister chromatids that separated down the middle. Now, those are big, big events, and all of those are going to be kind of connected here when we answer the question on why you're so unique with the help of meiosis. So first, let's talk about mutations for a minute, because ultimately species change over time on planet Earth. And ultimately, the bedrock of that idea is the fact that there's mutations on planet Earth. They happen all the time. Okay? In normal, healthy species, They there's a background rate where they do constantly happen, but it's often very long time. A long time in which will pass before there's any significant change in a species. But sometimes in simpler organisms like bacteria and viruses, those mutations happen a lot more readily. You can just use COVID-19 as an example. Since March of 2020, we've had multiple strains, anywhere between 5 and 10 new ones, new structures of COVID-19 that have emerged because it changes over time. It's an exposed little virus particle made of some protein and some nucleic acid. And it's out in the sun, it's getting hit with chemicals, it's outside of the body, and sometimes in the body when it changes. And it's very normal for a virus to change like that. Okay? Amongst other more stable organisms, those changes may take longer. But when we're talking about meiosis, it's not so much mutations that we're going to be zeroing in on, but instead the reshuffling of alleles during sexual reproduction. So if you remember a couple talks ago, we mentioned the word allele, and we said it was alternative forms of a gene. So let's say we had a gene that controlled eye color. Alternative forms could be a blue eye color gene, a brown, a hazel. It could be a number of different possibilities for eye color shade. Okay? So that's an example of a set of alleles for a particular trait. And what we're going to do with meiosis, we're going to shuffle that all up. We're going to make new combinations that have never been combined before every time we make one of these sex cells, a gamete, sperm, or an egg. Okay, so the behavior of chromosomes during meiosis and fertilization is responsible for most of the variation that arises in each generation. And there's 3 biggies for this, okay? Number 1, independent assortment. We'll dive into this after this slide, but this is the one where we said during metaphase, they all line up down the middle. And, what are the rules for lining up in the middle? Are there any rules or not? And that is definitely going to affect the amount of possible outcomes to the meiosis process. Number 2, crossing over. So in that stage in prophase where they find their partner and they're floating with them, they exchange that information, that lends to a great amount of genetic change because you're swapping portions of chromosomes from one to another. And then third, we have something called a random fertilization. And that is going to center around the fact that every time you make an egg or every time you make a sperm cell, they are very, very different than every other one that's produced. And there's mathematics behind this and probabilities, and you'll see how much you are unique, both qualitatively and then numerically in terms of, we said there's that phrase, you're 1 in a million. By the time we're done, you're going to see that you're much more unique than 1 in a million. Okay? So independent assortment, crossing over, and random fertilization are our big 3. Okay, so let's start with independent assortment first. And I drew a cell up there that's in metaphase, and I just drew the paternal chromosomes, okay? So those are the blue ones. So what I want to do, let me get my red going here. I'm going to use red for the maternal chromosomes. Let's give that a try. Okay, yeah, so over here on this side, I'm going to put the opposite one. So red will be the maternal chromosome of the pair. And you can see, completely random on how that lines up. Remember, there's 23 pairs, okay? 23 different pairs of maternal and paternal chromosomes that are going to line up down that central metaphase plate. Okay, so the combinations possible when chromosomes independently assort is going to be 2 choices raised to the number of chromosomes there are. So for us, we have 2 choices and we have 23 pairs. So if you crank that number through 2 to the 23rd power, it's 8 million. There's 8 million different combinations of how to arrange the chromosomes down the middle. So already, without even crossing over in random fertilization, you're already not 1 in a million, 1 in 8 million. Okay? If, let's say we didn't have a 23 pair cell, we had a 2 chromosome cell. Okay? There'd be 2 ways to set it up at metaphase 1. We could put the paternal ones on the one side and maternal on the other. And then in the other cell, you could mix and match, one of each. And just with 2 chromosomes alone, you already have 4 different choices, 4 different gametes, sperm or eggs. If I had 3 chromosomes, then I would be up to 8. If I had 4, I'd have 16. If I had 5, I'd have 32. And you can see how those numbers start taking off and skyrocketing the more chromosomes you have and the possibilities of gametes. Number 2, crossing over. Crossing over produces what we call recombinant chromosomes. Now, why are they called recombinant chromosomes? Because their genes and their sequence of DNA has been recombined into something new. So they are brand new arrangements of DNA after that crossing over occurs. Again, if you forgot what it is, the 2 middle chromatids of a homologous pair, they're called non-sister chromatids. They're going to exchange information like you see right here. They're going to cross over each other. And when it's time for those 2 homologous, for the homologous pairs to separate at anaphase 1, notice what happened. The segments have now been added to the other non-sister. So in this case, the yellow one has a purple foot, and this purple one has a yellow foot. That sequence is now new. It has been recombined into a brand new order. And then once meiosis continues and separates, those chromosomes and chromatids have all new arrangements headed for new cells. So again, if we go real simple here, you can see if we have just a single pair of homologous chromosomes and they cross over here at the chiasma, you can see as a result when the cell splits, you got the blue one with the red hand and the red foot, and you got the red one with the blue hand and the blue foot, if we could call chromosomes having like body parts. And then when they do their separation, the sisters come apart, and what do I get? I get 4 different possibilities. An all blue one, a blue one with the red tips, a red one with the blue tips, and then a red one. So even with 1 pair of chromosomes, I already have 4 different possibilities and outcome with 2 recombinants in there. And number 3, random fertilization. So understand that during meiosis, millions and millions of sex cells can be made, and each one is going to be a result of the process. So with that understanding, any 2 egg and sperm can meet at some point, okay? And we already said that each one of them is sitting at 8 million possible combinations already due to independent assortment. But what happens if I take 1 sperm, 1 of 8 million, and 1 egg, 1 of 8 million, and then I put randomly those 2 together? Well, in math, what we do is we multiply those 2 numbers. So 2 to the 23rd power times 2 to the 23rd power is 70 trillion. Okay? So you're not 1 in a million, you're not 1 in a billion, you're 1 of 70 trillion possible combinations. That's pretty unique as an organism to have that much variation possible, and you're the product of it. Okay, any questions there on why you're unique through meiosis? All right. So the process is very good. Most of the time it works out just right, but like all things biological, sometimes it does not, okay? So what happens if meiosis doesn't work out right? And here we're gonna hit learning target 2, 3, and 4. Number 1, the term euploid means you have the correct number of chromosomes in a species. For humans, it's 46. Like we saw a couple days ago, a potato is 48, goldfish is 94, that's the correct number. But as soon as you deviate from that number, 46, and say just one more, 47, 47 would qualify as being aneuploid, okay? It is not the correct number that our species is supposed to have. It means that there was a mistake somewhere along the way. Now, in some living things, it's devastating. In others, it is not so bad. Like in the plant kingdom, aneuploid events happen a lot, and plants just seem to kind of roll with it. Like bananas, they're triploid. Strawberries from the lab, they're octoploid. So they're cool with changing up that, and they just kind of roll with it and continue on. But animals have a harder time with changes of chromosome number.
Staff and students, will Rita Chowdhury please come to the main office? Rita Chowdhury to the main office, please.
And these problems typically result from something called nondisjunction. Okay? Nondisjunction. It's a failure to separate properly. And when that happens, we typically see two outcomes. One is called monosomy. Okay, monosomy, the prefix mono means one. So where you're supposed to have two chromosomes, there's just one there. And think of that as like not enough instructions to build something and run something properly. And then there is another outcome called trisomy. And the tri in trisomy stands for 3, which means that you have 3 chromosomes instead of 2, which means there's like too many instructions. There's too much happening that's trying to get the cell to do certain things, and the cell's a little confused, and it makes too many products. So nondisjunctions, I want to run you through a couple of scenarios that cause them to happen. There's two. There's one called primary, and there's one called secondary. Now that primary and secondary term refer to whether it's meiosis 1 or meiosis 2 where it happens at. Okay? So let's run down a primary nondisjunction first. So in this one here, you see a pair of homologous chromosomes, and this is right before, this is like G2 right here, and it has to then produce some sex cells. So normally what happens, it goes through prophase, they find each other, they line up down the middle, and then comes anaphase, where they're supposed to separate one chromosome per cell. And as you can see, that did not happen. Instead of one going into each cell, they both went into this one, which left this one here empty. This egg cell is empty. Now, during meiosis, the process will just continue on even with the error. So these two cells here will go into meiosis 2, run through the same actions that they always do, and then when it's time for anaphase 2, for that to roll around where sister chromatids separate, you can see they did, but you still have 2 chromosomes per egg here, and these two separate and there's none in there. Now let's imagine this is the egg that gets fertilized. If a sperm carrying its own chromosome joins into this egg, and this one and this one and this one, what do you expect? Well, if there's already 2 and you're adding one more in there, you get 3. So that happens in both of these cases here. So these two are what we call trisomy. Because of a failure to separate homologous chromosomes way up here during anaphase 1, now these 2 egg cells have too much genetic material in them at one of the chromosome positions. These over here, if these get fertilized and it's just the DNA from the sperm head going into the egg, there's just 1 chromosome. There's not enough instructions for a normal cell to do what it's supposed to do. We call that monosomy. Now, in a case of secondary nondisjunction, same thing, got G2 going on here, it's about to divide, so it goes through prophase and metaphase, and then the 2 are successful and go into separate cells. This one goes this one goes this way here. But then in the second round, it jumps into meiosis 2, and here's where you're supposed to have the sisters separate. They do fine on these 2 cells on the left, but they don't do fine here. The sisters right here hang on to each other, and they both wind up in one of the cells, and 0 sisters end up in this one. So what do you have as an outcome? Well, in this case here, these are actually going to be normal, because right here the sisters separated just fine. The sperm fertilized them, and you have 2 normal cells here, even though there was a non-disjunction event. But some of the cells will be affected. For example, in these 2 here, this one, if this is the one that gets fertilized, will again be monosomy, and this one here will be trisomy. So 50% wind up being normal, 25% here trisomy, 25% monosomy, 25% chance. Okay? Now again, monosomy, without enough instructions, the cells usually don't last long because there's just not enough to run the cell metabolically, physiologically. And with trisomy, the cell is forever affected because there's too many instructions, the cell's confused and making too much product. So ultimately, this first cell, keep in mind, it's now going to start doing mitosis while it builds a brand new organism, whether it's a person or a dog or a salamander, whatever, with the wrong number of chromosomes. Okay? Now, yes?
Is it possible for it to mix up on both primary non-disjunction and secondary?
Is it possible for 2 non-disjunction events to happen at once?
So like one in this cell for one of them to have
It would be possible, like in the primary case, if they didn't let go at all and they all wound up in one, yeah. So you have 3 monosomic cells. You have one. I've never heard of a pentasomy scenario, but I suppose it's possible. And I would imagine it would be absolutely disastrous for if it was an animal. If it was a plant, who knows? It could roll with it. If it had some cool new traits to make it survive a tough environment, it could happen. Good question. Okay, so what does it look like when it happens in us? So here's one of the examples of trisomy occurring and what happens physiologically, metabolically. So the first thing I want to ask is, if you look at your sheet and you look at the karyotype, which is where they lay it out on the black background, can you notice a place that isn't quite right on the karyotype? Can you find it where there's a mistake? Andrew, what do you see? What's that? Yeah, if everybody check out position number 21 right here. So 21, the 21st set of chromosomes should have 2, but they have 3. And this is what we call trisomy. And what this results in is what's called Down syndrome. How many have heard of Down syndrome before? Okay, quite a few of you guys. Okay. So by having that extra chromosome, it again will make extra products. It's additional instructions that normally diploid cells don't have to try to figure out. And the outcome of it, you can see that in a list there. There's some physical things that happen. There's some mental things that happen with having that 3rd chromosome, metabolic things as well. And the number is higher than a lot of people know. So if we just flat out took the overall rate, it'd be about 1 in 700 births have a child with Down syndrome. But I want to add this little extra over here. It's kind of hard to see. It's kind of small. What this is it's an age scale for the incidences of Down syndrome. Now, at age 20, which is a young reproductive age, okay, not the youngest it could be, but it's fairly young in today's society, about 1 in 2,000 births would result in Down syndrome. At age 30, the next column over, about 1 in 900. Okay, so you're going from 1 in 2,000 to 1 in 900. By age 40, it's 1 in 100. You go down to the bottom right, at age 49, it's 1 in 10 births result in a child with Down syndrome. Any thoughts on why that could be? Like, why would it, as you get older, would the chance of Down syndrome increase so much? Can you think of any reasons? Okay, so when you're younger, everything seems to kind of work a little bit better. We usually, gain the illnesses and the health problems a little bit later in life. But I also want you to think about the eggs. The eggs have been there since birth, right? So a woman, a female, is going to be born with all the eggs she'll ever have. At age 20, they're only 20 years old. But age 30, they're 30 years old. Age 40, those same eggs are 40. Age 50, they're 50 years old. That's a long time for an egg that's halted in the middle of meiosis to just hang on, freeze, don't change. You know, we do a lot in our lives. We're exposed to a lot of things. And by the time someone's 50 or 60 years old, a lot of those eggs have some issues with their chromosomes. All right. What about those like that was an autosome, but what about sex chromosomes? Can they change? Could that happen with them? The answer is yes. So their changes will result from a lack of X's or Y's. I'll give you two examples today. So a nondisjunction let's say a sperm was produced and it didn't have an X or a Y in it. Then if it fertilizes the egg, what happens is she'll get the X from the egg, but there won't be anything else to join with it in that 23rd position. And this will result in something called Turner syndrome, which would be we call the genotype XO, the genotype of the letters that represent what she has. She has one X and then nothing. So ultimately that will affect, again, physical traits. It will affect her ability to reproduce later herself, because she's missing that other X. So she just has what's on the one and without the other. Now, men, on the other hand, they could possibly receive a trisomic condition in their 23rd position, which would be like two X's and a Y. So if there were two X's in the egg and then a sperm fertilizes it, and it has a Y, it is a boy because there's a Y there, but there's an extra chromosome again, which means too many protein products are going to be made. The instructions are going to be confusing to the cell, and overall, since that same cell will divide, hundreds of billions of times to make an adult, all the cells will have that same difference in it, and it will lead to those physical traits that you see there. And again, physical and future reproductive capability, usually both of these will result in people being sterile, which means they can't actually have their own kids. They could adopt kids if they want kids, but because of this chromosomal abnormality, it's just not possible. Okay, and if you want to take a look at what that looks like on a karyotype, if you look in her 23rd position, you see the X, but you don't see anything with it. It's just missing. Okay, so this would result in Turner syndrome. He, in his 23rd position right here, has an X, and a Y. So he has that third chromosome in there, extra X, which is going to lead to those conditions you saw in that prior slide.